Now we continue and discuss clinical immunology part 3. And some questions. And so antigen recognition by T lymphocytes is going to be looking at what goes on outside of the cells. How T cells are seen antigen. And then we discussed how things go on inside. Question to consider. How do T and B cells see antigen? How does T cells distinguish a foreign antigen or infected cells? Why do some T cells kill and others provide help? So macrophage originally have two functions, phagocytosis and antigen presenting cells. Dendritic cells have to function also, uptake antigen in peripheral organ and antigen presentation. Macrophages and T cells activation macrophage. Macrophages have a lot of receptor on surface membrane. First of all, present MHC, major histocompatibility 1 type, major histocompatibility 2 type, receptor toll-like receptor we mentioned before, receptor to antigen as Fc fragment antigen and adhesion molecules. T cells activation macrophages, it's important role for T cells through CD4 receptor and 3D4 ligands on lymphocytes, resulting junction between each other, forming activated T lymphocyte is producing interferon gamma. Interferon gamma released from T lymphocyte and more effectively attach and activated macrophage. Macrophage also activated and compare between major histocompatibility complex 2 type and bound peptide through bound peptide. Antibody. We mentioned about construction antibody. Refreshing only this topic. Have FAB fragment to binding antigen binding site. And variable part have a light chain, have a heavy chain structure, and Fc fragment this activated another complex as a complement complex, macrophage, T cytotoxic cells. B cells don't have to be anywhere near the infected site. Molecule have two functions as antigen binding site. Depend which FAB site have this molecule. For example, immunoglobulin G have two binding site. Responsibly, they could bind only two antigen. Pentamer as immunoglobulin M have 10 FAB fragment and binding 10 antigen, and respectively another immunoglobulin A and immunoglobulin E. T cells have to get to where the infection is. It's a very important notion and different between T and B cells. The T cells receptor only recognize antigen in the antigen presentation cells. And scientists needed to know T cytotoxic cell T cells plus antigen don't go to no response to immune system. Only T cells plus antigen and plus antigen presentation cell may be able to act as immune response. Conclusion is antibody and T cells recognized different antigen. T-dependent and independent type activated B cells. B cells production, of course, immunoglobulins, but without activated T cells, and compare with mitohistocompatibility 2 type, B cells produced low affinity antibody as immunoglobulin M. If this disease or antigen dependent T helper present into this reaction, T helper compared with mitohistocompatibility 2 type at activated B cells, and this produce immunoglobulin high affinity as immunoglobulin G through activated T helper and production a lot of cytokines. Now we discuss about scenario to response and defense from tuberculosis. Okay. Tuberculosis bacteria is going to be unique to other bacteria. They rod-shaped saurians, similar shaped another bacteria. What is the unique in bacteria tuberculosis? It has a special coating, is made up mycolic acid coat. So first of all, you can see it's very thick waxy coat. What this waxy coat does? Increase resistance against the immune system, so it's more difficult for macrophages to break it down. How is this also slow down the growth of the bacteria? Normally bacteria grows 20 minutes. However, this bacteria grows in between 15-20 hours. So although you get higher resistance because it have this coat, you know the downside is that the bacteria will grow much slower since it has that coat. Gram stain is a little different actually in other bacteria stains. Also Legionella, for example, Cryptococcus sporidium. Finally, these bacteria don't need a lot of oxygen, so they are aerobic. Let's talk about pathogenesis. So, in order to get infected, the first things that need to be exposed. Bacteria has to, something floating in the air, probably from another person coughing up, has to make its way through the bronchial into the trachea, bronchia. It needs to go into the lung. Here, how now the place where we pointed around the upper lobe and the lower lobe of the lung. And primary infected tends to prefer lower upper lobe in lung. What happened next? Macrophage will phagocytosis it. And in other words, they will eat it up, and the bacteria inside macrophage, within it will be a formation phagosome. Another thing here is lysosome is full of acid. What needs to occur? It's phagolysosome. Mycobacteria release certain factor which inhibits the fusion of lysosome with phagosome. So this doesn't occur. So what happened instead is over time mycobacteria starts to adapt. Finally, phagosomes ruptured. Macrophage ruptured to release mycobacterium outside. What is going to happen? Macrophages will eat up all of this, and so mycobacteria evade phagocytosis. Thrive mycobacteria infected, where most bacteria would die. What will happen eventually is you get a granuloma. Earlier, you have a lot of mycobacteria with mycobacteria in there, and over time the anomaly of macrophages will higher. Cell-mediated immunity forming through mycobacteria, completely surrounded by lymphocytes. These macrophages activated T cells. T cells activated release lymphokines. Lymphokines activate macrophages again and completely surrounded by lymphocytes. This activated macrophage release lymphokines a lot of number and central these areas, the surrounding lymphocyte and central have mycobacteria, and over time this forming central necrosis. Necrosis macrophage kills this tuberculosis bacteria. Information special type necrosis, caseous necrosis, or means cheesy necrosis. This pathognomonic necrosis characterizes only Mycobacterial tuberculosis. It forms primary tuberculosis. Pretty much debris macrophage plus Mycobacterium tuberculosis. Not all are killed. Sometimes Mycobacteria remained dormant a lot of time. After this, 90% of people primary tuberculosis would not progress any further. What is characterized as primary tuberculosis? Sometimes is asymptomatic and sometimes have something vague symptoms as a chest pain, fever, erythema nodosum present on the leg. Erythema have not only tuberculosis, have another disease maybe accompanied as a sarcoidosis, histoplasmosis, and sometimes applied to primary tuberculosis conjunctivitis. After primary tuberculosis, forming scar into the tissue, lung tissue, and maybe tend to progress this disease. You know that if a child in child and HIV patient, immunocompromised patient, this results in large granuloma forming cavitation. This cavity could get infected, go into the pleura, effusion 2/3 of time, penetrate pleural space, enlarge lymph nodules, compression trachea, bronchoectasis, and hematological spread in other organ, internal organ, and disseminated tuberculosis forming. 10% go into lung into peripheral part and forming gone complex. Gone complex at called complex as lung in peripheral part and involved lymph nodules or trachea, forming gone focus lymph nodules. And plus hypersensitivity reaction may be occur because Mycobacteria tuberculosis tend to 4-type reaction, hypersensitivity reaction occur. Later it can reactivate, this called post-primary tuberculosis or secondary tuberculosis, and located upper and posterior lobe in lung. Important to distinguish because of the fact that aerobic. Which signs characterized this 10% in other tuberculosis? Signs is fever, night, sweet, cough, non-purulent earlier, however, as time purulent, can even be blood. Respiratory distress forming once third patient have chronic consumption throughout whole life. Diagnosis test: chest X-ray, acid-fast bacilli microscopy, up to microscopy, culture test, as this picture, culture test. Next, Nelson stain, PCR reaction, but this is very expensive, and quantiferon test. Recently discovered this new test. We found present antigen from Mycobacteria tuberculosis. Next part we discussed some later.